saying it. They told me to shut the front door. Oh, thank goodness. Okay, I'm going to start. You're going to be quiet. Chatterbox. I know it's great to be back on site, but give me a break. All right. Nevertheless, happy we have people. We have visitors online. Hello to everybody, and I will begin. So let me get over here just to show you. I did. If you and I don't know if anybody, even I don't know if anybody had a chance to listen to what I put together on Monday. But and you should listen to the recording because I talk a lot about the background of what microbiology was historically, starting with ancient Egypt and ancient Greece and ancient Rome, all the way through what we call the Dark Ages, and they were dark for a good reason. And because it was really an age, and I'm talking about Western medicine, not Eastern medicine, which I'm really not qualified to talk about. And I do like, and I will use a lot, I'll be flip-flopping between PowerPoints, the Cowan PowerPoints, which you can see here. And I better scroll in the right direction. I'm scrolling in the wrong direction. And here, this Anderson, which is what I call Nestor, because if you look at the PowerPoint, you'll see it's Nestor's Microbiology. Anderson was just the most recent lead author on the most recent revision, which is this. So I'm going to use. I like the illustrations and the arrangement of this PowerPoint as well as I like Cowan. They're both McGraw-Hill. They're similar but different. And so I happen to like this one particularly for the historical figures and some of the things that we'll be presenting today. To begin. So give that a listen when you can. It's important. I don't want to go over it, but I will have to reiterate, or I'll have to at least cover some of it. I don't know if that's the right term. But you can see it here where we say Nestor's Microbiology. That's why I call it Nestor. And I did go through a lot of these things that were, and many of you in the lab already, I understand if you had lab yesterday, as some of you did, will have had some of this material. And this is more lab than anything else. So I'm not talking about this. You saw this under the microscope, I understand. Understand that the whole idea, the premise of what today's lecture about is understanding what preceded this. And why I ended the thing I did on Monday, the recording I sent you, it really ended with the work of Pasteur. The whole idea was Pasteur came up with germ theory, which is today that tiny things we can't see, microbes, microscopic particles that we can't see, can cause disease and cause other problems. And now we know they're for good or for bad, whatever. So it's germ theory. Prior to that, he refuted relatively efficiency, but not without incident. What was the theory from the Dark Ages, from the end of the fall of ancient Rome, all the way to basically the Renaissance, and even continuing through the Renaissance for about 250 years before it finally was accepted, something called spontaneous generation. Another name for that, all one word, because A means without, biogenesis. Something to grow biologically, that basically growth occurs without some biological impact. Like magic, the chair is going to come alive and start running around. Okay, and it's just like you see in a sci-fi movie. You see, the witch or the wizard, boom, and all of a sudden things start moving around. Very Fantasia. That's a Disney reference. Okay. Anybody know what I'm talking about with Fantasia? Back in the day, we used to watch that. That was very influential. But I digress. So, effectively, that's kind of the frame for today. What this is all about, how it's impactful to us, all the things that were directly or indirectly brought to bear by Pasteur, and why he had the most credibility. So, I mean, and I'm not going to ask you about who did the microscope, and this was a single lens, and that's lab. But the stuff that von Leeuwenhoek was seeing, who was a fabric merchant with a little tiny single lens microscope that magnified 300 times, you see a picture of stuff you're seeing in lab. Cocci, Spirilla, bacilli, rods. If you didn't see them, you will. You'll get sick of seeing them. You'll see so many. So, I mean, this is the early days, even before this, historically. So here's that little magnifying thing. And I went through these slides a little bit in the earlier. I'm not doing the thing. And microbes, we use the term that versus microorganisms because viruses and prions are acellular. And this was the whole idea. And it didn't take 200 years. It took a millennia, more than a millennia, maybe a millennia and a half, maybe 1,500 years to disprove. It was all, I mean, and this is not an indictment of church doctrine, but they felt that if you got sick, there was some evil force in the air, or you were in league with the devil, or that kind of something, some terrible thing, retribution from the Almighty for something that you did, that kind of. And the treatments were you couldn't, because it was church doctrine. And I'm talking about Western medicine as opposed to medicine, Eastern medicine, which would be Asia, Japan, which is very different. I'm not really qualified to speak about it. If you went against that, it was heresy. And I mean, you would be punished as any heretic would be, burned at the stake, tortured, etc. That's so. What happened in the early days of the Renaissance? That was the whole idea. We began to view and look for things science. All those wonderful anatomical diagrams came from cutting up dead bodies, which was, again, people would have been subject to, would be branded as heretics. And that includes people like Da Vinci and Michelangelo. That's how they were able to draw these things. Your ancient, and someone named Vesalius, who was from Italy, and all these wonderful original muscle diagrams all came from that. How did they do those brilliant sculptures and painting? You have to think about that. Well, and I came onto it, and this was the first one in the 1660s. That was about 200 years before Pasteur, and it still took time after Pasteur. He made it. This is science. And that was the change. He made an observation. He saw meat that was left out. They didn't have any refrigerators, certainly. Wouldn't need one today. See, two people smiled. The rest of you are still stunned. Thank you. And he saw flies lighting on it. And the flies, when they left a little bit while after that, he saw maggots coming out. And he said, the flies must have been laying eggs. It wasn't some evil force in the air that magically made the maggots occur. And he conducted a simple experiment. It wasn't 2 jars, it was 3. And he put one in a glass petri dish, just raw meat. He put one with a glass lid on it. Air couldn't get to it. That was a big deal. And then he had another one where he put cheesecloth. He was Italian. They used cheesecloth. And he reasoned that if I put a layer or two of that, the eggs from the fly couldn't get through. And he was absolutely right. Except they said, that cloth is not, well, the air couldn't get to it in one. That's why it didn't spoil. And now you got that cloth on here, and it's pushing away that force in the air, that life force in the air that could create things. Really? So and again, it wasn't acceptable. And understand the concept of validity or validating means that you and I could do the same thing in our lab and come up with the same results. That's the problem in practice Pasteur, as you will see. And what happened to that? I basically, that's all materials and methods. You're doing a lab report. I can do the same way. Well, people tried that, but if they didn't have the exact same cheesecloth that was small enough to not let the eggs get through, you couldn't duplicate it. That's the idea. In science, you have to duplicate something exactly. And so you got to look for you try to have just one variable that you can control in a scientific experiment. That's just me giving you some background. So a lot of people didn't understand this concept of spontaneous generation. And as you're learning in lab, how do you grow stuff? You grow stuff either on agar plates or you use broth. And broths are just that. They're nutrient broths. They're made out of a variety of things that are dissolved in water, and they're sterilized. How do we know that they don't have growth in them? They're a solution, which means they're clear. Okay. Solutions are clear. When solutions get murkiness or cloudiness or little strandy things growing in them, we call that turbidity. We learn about this in the lab. We can even measure it to see how much growth there is with a spectrometer. You may even do that in the lab. We don't generally do that in our labs, but it's been around forever. And they do it in hospitals. And what's interesting about that is you're going to see that broths play a pivotal role when we get to Pasteur. So everybody was trying to figure out a way to debunk this concept of spontaneous, because by that time science said this is just can't, and we've already had a microscope and people are looking around. So he tried boiling broths, but they still produce microorganisms. And you're going to see all broths are not created equal. Somebody else boiled broths longer, sealed the flasks, air couldn't get in there, and broths remained sterile unless the neck was cracked. But the question is, did heat destroy the vital force of the air, and blah, 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 blah. And nobody could came up with it. And enter into the fracas, and you'll see I commented, and I should have mentioned to the other class, I have a handout on historical figures that you must look at. Because you better learn about Jenner, because I talked about that on that thing. There was a start of vaccines, smallpox. This was before, 60, 70 years before Pasteur. He was saying that vaccine is so successful, we eradicated smallpox, to our knowledge, in 1977 because of a very successful vaccine that he took a major risk to produce. And smallpox is the ultimate biological weapon. Spread respiratorily, kills in about 3 days. Easily spread. Oh, believe me, there's still smallpox vaccine being held in storage just in case. Yeah, the military has a lot of stuff that they're very aware of, the old just in case. You end up in the military because you have a whole lot of shots are going in your arm. Oh. My eldest was the first one to go to work with the Cambodian Navy after the Vietnam era to set up maneuvers with our Navy. And he goes, yeah, my arms never hurt so much with how many shots I got going there. Semmelweis, you're going to hear how being a scientist isn't always rewarded. Because he became scorned and shunned because he published an article showing how doctors were responsible for puerperal sepsis, childbirth fever. Please listen to that lecture. I should have made that point to the other, and I will. Which brought us to Pasteur. Shortly after the Civil War, Pasteur had credibility. They knew he was very good with therapeutic. He understood the microscope. He used it. He always looked for ways to treat people. He wasn't so much a researcher as he was a doer. He was the first one to coin the term before he even knew about a virus. He was the first one to treat the dreaded disease of rabies by extracting fluid from bloodstream or tissue fluid from animals that had rabies but had not succumbed to it, in an effort to basically make the very, very early treatment of antitoxin, of antibodies to fight rabies. He didn't know that he was doing all that, but he was one of the earliest ones that had success treating rabies by animals who had had rabies and appeared to have recovered. The reason there was something in their bloodstream that he could extract to inject. So he was all about that. I mean, that's really remarkable to be able to find that. And it sort of was following the lead a little bit of Jenner, who I talked about with smallpox. But when they knew that, and what happened, he was courted. He was not courted. He was basically hired by the wine industry. Wine was a big industry. And wine was spoiling like crazy. They'd bottle it, and it had a very limited shelf life. It was cast at a very limited shelf life. And they were losing money. They go, what's going on with the wine? Well, when wine spoils, you ferment wine basically with yeast and other microorganisms. Fairly simple ones. And if you don't do something to stop them, they will continue to ferment. They may be drinkable initially, but when you continue fermenting, you stop making alcohol and you start making vinegar. We even use the term vinegar from the Latin word vin, wine, agre, sour. Sour wine is vinegar. It's desirable if you want to buy red wine vinegar or balsamic vinegar or white wine vinegar. That's fine if you're cooking, but not so good if you're trying to consume expensive French red wine or white wine. So they had a big issue with that, and he reasoned when he took samples, he saw some of the microorganisms he had been studying because he'd take little pieces of cotton, saw what grew on them. He was experimenting with broth and flasks, as you'll see. And he said, if I can heat these strategic, because if I boil it, the wine's going to lose what makes it wine. it's not going to be basically a commodity that's worth anything. So if you're testing it, he came up with this, roughly 62.5 degrees Celsius or 140 degrees Fahrenheit for about 30 minutes. And if he would hold that, get it to that temperature, they had plenty of thermometers for about 30 minutes and let it cool naturally, it didn't spoil. It didn't spoil in the cask, didn't spoil in the bottles. And we have a name for it. We named it pasteurization. It's not the only way. It's not the only temperatures and times that we use today. We get to that in a subsequent chapter, probably in our second major unit. We look at what's called methods of control. We pasteurize everything you consume is pasteurized. If you did not know it, your dishwasher is pasteurizing. When you put your dishes in the dishwasher, roughly it reaches a temperature. Just look at the specs: 130 to 140. It keeps it there. That heat's there for about a half an hour. That's what you do. If you didn't know that. Pasteurization doesn't get, it's not sterilization. It doesn't get rid of all the microbes, not even close. But it typically, the ones that you consume that could lead to foodborne illness, it gets rid of the vast majority of them. So you're going to hear, and we'll talk about things like ultra pasteurization, which is totally different. Things like that down the road. But this is just the primer. That's his credibility. And he did, and I'll get back to the PowerPoint and show you what he did. It's one of my favorite lectures, if you haven't guessed. I wanted to do it when I had a live audience. That may be doing something. So he basically knew about microorganisms. He trapped them. He found them, and he looked in the broths that they were having trouble that we're working with, and he reasoned that this cotton plug, when he put it into broths, I mean, they got contaminated easily. So the whole idea was to be able to prove or to disprove that theory. He had to leave what was sterile broth exposed to the air and show how it would not become unsterile by becoming cloudy or murky. So he used a broth that he boiled, reasoning from previous tests, and he had the glass blowers create a swan neck or gooseneck kind of a stem that was open. And he reasoned that any of those particles would go into the bend of the neck. He was absolutely right, because before he had experimented and had little bits of cotton and trapped them, so he knew they were coming there. And once that was allowed to cool naturally, it stayed clear, sterile, and not just for a day, but he had some for a couple of years. And whenever if one would tip over, contaminated. If the stem would break, contaminated almost immediately. Hours and days, boom, murky and cloudy. Great experiment. He did. I mean, it was very, very useful. Big problem. People couldn't replicate it because the broth he used was from stuff he was familiar with. He used a sugar and yeast containing broth. It's like you got brewer's yeast or baker's yeast and put it out there. Nutritional yeast that we use today. Okay? And use that with sugar to make his broth. A lot of the other broths were using what's called hay infusions. An infusion is like a cup of tea. You take typically some kind of plant material, you put it in heated water, maybe you boil it and then you let it cool. And when it reaches a certain temperature, like tea, about 190 degrees Fahrenheit, it extracts. That's the optimal temperature. Our British friends are very good at that. Okay. That's what an infusion is. They used hay infusions. And the difference was this. They couldn't reproduce it. They couldn't validate it. Okay? Sterilizing broths required different times, some in 5 minutes, others hours. And believe me, everybody, what you're going to learn in this class is that boiling is not the way we sterilize. Because there were, particularly in the hay infusions, they had heat-resistant microbes, which very important name going forward, called endospores. The definition of sterilization is the removal of all microbial life or all microbes, including bacterial endospores. If you can do this, you sterilize. We don't boil things to sterilize them. Most of the time, we use a pressure cooker. typically in the hospital. We'll go over the different means of sterilization. The most common one we use in medicine is basically, you could do it at home with your Instant Pot if you have one, or any type of pressure cooker. Easy. It's called an autoclave. We'll get into all the numbers. Effectively, they superheat it under pressure, and it kills the bacterial endospores. If they're gone, we know everything else is gone. We'll get back to that definition down the road. The problem was, is this hay infusion? They would pick, after hay was picked from the farm, it was grown, baled, you had little bits of hay that was lying around the ground. They pick it up and they make the infusions from that to try to do these experiments. Well, it's got, in the soil, it has a whole lot of microbes, particularly bacterial endospores. And understand, everybody, I'm going to tell you this now, it may surprise you. In the carpet, there are bacteria, they're effectively indestructible. They live not for 5 years or 2 years or 3 years, but hundreds, thousands of years. We only can use it. The only thing to get rid of them are extreme methods like high temperature, powerful radiation, powerful chemicals, etc., etc. They're in the bottom of your shoes. They're in the carpet. They're in your home. They're in your dorm room. They're all over. So much so that we always ask about them. And there's always a danger that they will get into a wound. If you go to the emergency room or an urgent care, this is one of the questions they have. You have an open wound. What do they ask you about? And I know they ask you about the most important thing to them, your insurance card. They'll ask you about allergies. They'll ask you about medicines. They'll ask you about who your doctor is, if you have one. What do they ask you if it's a wound? They'll ask you about some recent treatment you might have had and how long ago it was. Oh, come on. The other guy's got it. Your buddy got it. I gave them a hint. It had to do with a recent form of preventative treatment they might have had. When was your last? You're killing me. Tetanus shot. 20 lashes with a wet noodle. For all of you. When was your last tetanus shot? The name of tetanus, everybody. Clostridium tetani. Any wound, potentially, and we always use the genus, is always capitalized, and we typically underline or put in an italicized letter, and the species is always small. And we use binomial nomenclature, so all you have to do is Clostridium tetani, and we know what it is. It is an endospore bacteria that happens, why it's so interesting, it's an anaerobe. It has to grow in the absence of oxygen. It won't grow, oxygen kills it. And so what happened is in any type of a wound, basically, if you go to the emergency room, if it's the vast majority of wounds are typical kitchen cuts, they need a couple of stitches. They're not tetanus-prone wounds. Tetanus-prone wounds are really bad wounds. Like in the backyard, you stepped, and it's more than just a stepped on a nail, even though that would certainly be an example. Man versus lawnmower, bad injuries, car accidents, stuff where bone or tissue is exposed. And particularly if it's irregular, because what happens is it interrupts the blood supply and it necroses, it dies. When something necroses, it becomes a breeding ground for anaerobic bacteria because blood with oxygen can't get to it. We call that a tetanus prone. So a lot of times when you're doing a tetanus shot, you want to make sure they're tetanus current. Okay, and we'll go over the criteria we talk about wounds toward the latter part of this class. And additionally, it's for the next time. It's for when they're out hiking and when they fall and hurt themselves. And they don't get medical care very quickly. Before things get started, you get 3. If you did not know it, there's 3 immunizations at once, trivalent. Tetanus, diphtheria, and pertussis, which is what we call it. And we'll talk about diphtheria down the road. And all of those are scary, potentially. They're all life-threatening. So without getting into the whole wound thing, it's all about that. We have to be concerned, and very rarely in surgery, but the first thing they tell us in surgery, if you have a wound that got infected, if you did surgery and got infected, then you have to start in the back of your mind, start thinking about it. So I thought one case, I'll tell you about it. I'll tell you about it again. One case of active tetanus that we saw, and it was missed in a small hospital that I trained at in Philadelphia, was in a homeless man. They should have suspected it. We all missed it. And it didn't come from a wound per se, it came from frostbite. He had to have his hands amputated. Frostbite, you know how they're homeless, out in the freezing cold. And it was more like February or March. They have those gloves that are, they have their gloves are all, their fingers are protruding. They all became gangrenous from frostbite. And nobody thought, none of us is wrong. I mean, was I actively involved in his care? No, but I was a senior resident. So all of us who were senior residents should have said, hey, make sure he's got 10%. And the physicians did too. It just underscores mistakes happening from the smart people, allegedly. Not alleged mistakes, allegedly smart. Okay, moving on. Another thing that you have to be wary about with these endospores is food poisoning. Most food poisoning doesn't bother you. You get over it. It's nasty, right? But, you get a couple of days, you have vomiting, diarrhea, and 2 or 3 days later, you rehydrate and you're good to go. One kind is potentially life-threatening immediately. It's very, very specific. It comes from a toxin. The toxin is something that I talked about to many of you in AP1 when we talked about exocytosis. We talked about a toxin that's so strong, we take teeny tiny amounts and use it therapeutically for cosmetic purposes. The name of that toxin is wrinkles and stuff. What do you inject people with? Thank God, somebody. A stranger among us. Its name, seeing a trend here, Clostridium botulinum endospores. You don't see it very much in war situations today, but the big problem in the Civil War, mechanized war, Civil War, World War I, World War II, before the chopper evac days and mobile surgical hospitals. I mean, in the old days, I mean, even in World War II, you had poncho and a couple of rifles and you carried somebody to the nearest field hospital. War wounds, explosions are the classic example, not just of tetanus-prone wounds, but they expose bone and they do a lot of tissue destruction. The thing that you were worried about in those days, because they've usually resulted in amputations, was a condition called gangrene. Yes, gangrene. And when we get to antibiotics, I'll tell you another anecdote about it. I'll review this. The organism involved, Clostridium perfringens. I have this on the handout, by the way. The reason we were so successful with World War II, part of the success is our soldiers had packs in the wound care packs. They had wound dressings and they had a green powder called sulfanilamide. When you sprinkle it on those wounds, the Nazis, the Japanese, the Italians did not have it. Only the Allies had it. Saved a lot of amputations. One of the big reasons, because that was a war of attrition. War of attrition. It was a numbers game in a lot of ways. I fancy myself as a little bit of a World War II historian. I urge you, if you're in New Orleans, go to the World War II Museum. It's nothing short of great. One of the best museums in the country. Love it. I could spend days there. My wife and I spent about 2 days there when we were in New Orleans. And the food isn't bad. Not at the museum, but the food's pretty good. So much jambalaya, so little time. Moving on. But these are all endospores. That pasteur, I mean, that's why boiling doesn't work. And we're not done yet. The one you're all going to see if you're in a clinical field, remember how we abbreviate them. We could call tetanus C. tetani. The one you're going to see, you can make the diagnosis with your nose as you go from room to room in the hospital. But see what? C. diff. Clostridium difficile, because it's so darn difficult to take care of. C. diff. We just nickname it. It's normal for a lot of us in our large intestine when you take powerful antibiotics for infection. This is going into my antibiotic presentation. It destroys the good bacteria. It lets the endospore containing C. diff, if you're there, the case, flourish. The reason the large intestine is an anaerobic environment. Almost done. So here we are. Another botanist identified these heat-resistant bacteria, named them endospores. The difference, pasteurs used broths of sugar or yeast, and the whole idea was the hay infusions, which brings us to the other really important character at that point in time. So, I have this on a handout, you will see. And that biologist and scientist was Koch. Pasteur, by the way, never won a Nobel Prize because he died before the Nobel Prize began, and they do not award them posthumously unless somebody dies after, when they initially announced the award before the ceremony. Koch did. Koch, where Pasteur was all about taking care of people, Koch, who was German and a little younger, was the lab guy. He did all the stuff you're doing in lab, plating things, which you're going to do, looking at the different kinds of augers, which you're going to use, and the different kinds of ways you grow things, isolating, particularly in pure culture. All about Koch. And he particularly identified an aerobic endospore that was causing a terrible disease, not so much in people, though it does certainly cause disease in people, but particularly in animals. It was killing animals and really affecting the economy, relatively agrarian, basically an economy that was based on animals for a variety of purposes, and as well as eating animals, wool particularly, and growing plants and things like that, called anthrax. Anthrax, big B, we use a small b, it's the shape of the rod, Bacillus anthracis. It's aerobic, and so you're actually not going to grow anthrax, certainly, but you're going to use one called Bacillus subtilis, which we finally call B-sub in the lab, to identify endospores and to do the stains for them, and that's what he did. He was all about that. He was the first one to grow in pure culture Staphylococcus, tuberculosis, Streptococcus, E. coli, etc., etc. He was all about that. And he did that for a really important reason, because he wanted to identify things. He felt it was really important. He developed something we don't use exactly, but similar today, about disease, called Koch's postulates. And we review them again when we look at how diseases spread in probably our third major. Basically, anthrax, they got sores. People can get it too. I haven't even started with that. They got sores, okay, and the animals were dying. And obviously, that's tragic, just from an economic as well as for the animals. And he took fluid out of them and he put it on a plate and grew it. And when he saw the colonies, like you're going to see in the lab, he extracted the colonies and he basically injected those colonies into healthy animals that he felt were susceptible to the same disease. Unfortunately, watched them die, saw the sores, pulled the fluid out of the sores, and grew the identical colonies again. It was like a chain of evidence. This is where they were healthy, they had the disease, I cultivated stuff from these diseased and dying animals, I injected it to healthy animals after I identified it in pure culture, and basically could do that continuously. And those were his postulates. That was how he felt diseases were caused. It doesn't apply as much today. We look more at the molecular biology, and there are certain exceptions. One of the most famous diseases of all, historically, can't be grown in pure culture, even to this day with modern techniques. The name of that disease is syphilis. So that automatically made this not a universal policy. There's a lot of different things, and I go into all that stuff historically. It's like one of my favorite things to talk about. When it gets to the diseases, as you probably can tell. I'm so happy to be here to tell you this. As much as I didn't mind necessarily being snowed in the effort of complete disclosure, I was so cold yesterday, I went long johns. It was cold. So, you want to go through this PowerPoint, you can look at this. These are all the major events that are there. Not all, but a really nice timeline if you want to look at it. We're not doing scientific method. I'll show you from the handout as we get ready toward the end of today's presentation what I'm talking about. So here's one of those handouts. Here's the historic figures, smallpox vaccine, Semmelweis, Lister. I should have told the other class, I'll try to remember. Lister took the word of Pasteur, the works of Pasteur, and he is the father of modern aseptic technique. Back in the mid 1800s, after our World War II, when they operated on people, they tied them down. They typically gave them a bunch of alcohol to drink if they could, and alcohol is a terrible anesthetic. Maybe in the later 1800s, they had some gases they could try to inhale that would produce a certain degree of anesthesia, and they started operating. They washed off the instruments. They didn't do necessarily anything to clean off the skin. They didn't do a whole lot of. Maybe they washed their hands. And they had a ton of infections. And he looked at that, and he started doing dilutions of a chemical called phenol, which is around, called carbolic acid. Even though we don't use very much of it today, it was the basis of something called Listerine, which was one of the original mouthwashes. Phenolic compounds, up until the last 10 to 15 years, antibacterial soaps, they were antibacterial because they contained phenol, but there's problems associated with it. And I'll tell you about that when we start looking at the different chemicals and their problems associated with it. And so Lister used these solutions that were safe. The physicians had to wash their hands, they had to clean off the person, they had to scrupulously wash and clean off the instruments, and he cut infections down tremendously. He was the modern of what we call aseptic technique. The term sterile technique is somewhat of a misnomer. Okay, so again, if they're telling you we're doing sterile technique, don't say, oh, Dr. Stein said that's a misnomer. Just kind of go with it, everybody. It's easy to take yes for an answer. So asepsis is what we practice, minimizing that. Sterile's fine until something is unsealed, then it's immediately not sterile. It's a question of how long before it becomes a significant problem. Here's all those Clostridia. You notice if you're going to write them for a report, not for me, but you either have to italicize them or underline them, etc., etc. Here's the last nugget for today. Doing good. Another one of these. It's genetic engineering I talked about a little bit. We really get into it more when we talk about bacterial genetics. We use yeast and we use E. coli because they grow so easily. We can grow large amounts, and many of them are receptive to genetic modification. They incorporate donated DNA comparatively easily. So, and we utilize these things called plasmids, these tiny circles of DNA that we can genetically engineer. We can basically splice them with sequences of DNA that we want, with the bacteria that we're going to engineer and alter to make things like insulin, to make things like hepatitis B vaccine, to make things like antitoxins that we're going to give to people, snake venoms and things like that, anti-venom, all of those things, and many, many more. I talked about Pitocin for labor and delivery. Relatively, I will talk about vasopressin. All of those, and you'll hear about that in endocrine, are relatively parts of engineering. And obviously we use it in the food industry. We've been doing things like this for years. Genetically modifying plants, whatever your philosophy is about them. And I mean, read up on it. And I mean, GMO foods versus non-GMO foods. I mean, if your budget allows, you decide what you want to grow. If you want to go organic and stuff like that, that's fine. So, a lot of that, I mean, we try to be cautious with that, but I'm not so worried about it when I'm in a hurry and I'm grabbing a burger at the Golden Arches. I know. Moving on. Gene therapy is interesting. I commented that in the recorded presentation because it utilizes a virus. The virus utilizes an HIV. Gene therapy is in its infancy. The difference between genetic modification is something we're doing in a microbe, yeast or E. coli, specifically to make a product. Gene therapy is trying to resequence the DNA of our cells. When we'll get to HIV, you're going to understand it's called a retrovirus. Retro means backwards. It's an RNA virus that has an enzyme that will rewrite the RNA into DNA, and it's a virus which easily incorporates the DNA into a host cell, relatively speaking. So we're trying, for instance, somebody with a cancer line, we're trying to maybe alter that line genetically or alter a line of white blood cells when we're talking about leukemias. We're trying to basically resequence those cells. And it's really in its infancy, and maybe that's down the road. That's going to be the nature of what medications are and what advanced medicine is. I don't know. But it's interesting. We basically take out the bad part of HIV, what it does nastily, so to speak, and utilize its ability to take RNA and rewrite it as DNA, and moreover, get that DNA to become a working part of a host cell, which it does. In our white blood cells, it's bad. If you did not know, what makes HIV bad is it basically attacks the single most important white blood cell in our body, called a helper T cell. It's the linchpin of our immune system. You'll be learning about that, many of you, in AP2 in immunology. I don't do a big immunology portion in this. I kind of skip those chapters because I have so much to cover, but I know you're getting it in AP2. Emergent versus resurgent diseases. Really important. The major factor for most emergent diseases, mutations. HIV is an emergent disease. As far as we know, it wasn't around until the late 60s in Africa. There's a lot of really good evidence to show that came from a failed, terrible, on the cheap, criminal attempt to make polio vaccine. That's why there's no vaccine trust in sub-Saharan Africa. That's why measles is still the third largest killer in there, because they won't take a very successful vaccine like MMR. They went to make a polio vaccine. They made labs in the middle of the jungle with dirt floors in monkeys. That was going to be their host to make this, to basically make the vaccine. They got any monkey they could capture. I don't know what term, which species or anything like that. They were so sick, they already had viruses, and the polio virus mingled with some kind of virus in there. I have seen that they're on like Science Channel. They have pictures of what these failed labs that were abandoned look like. It's horrible. Okay, that's a man-made mutation. Most of these are genetically unstable anyway. So bacteria, as you will learn in our genetics, are genetically unstable. RNA viruses are genetically unstable. That's why we not have to, but they make different flu vaccines every year. That's why COVID continually mutates. They're genetically unstable. They're RNA viruses. That's what they do best. The other thing is more than just mutations or resistance to antibiotics, which is genetic as well frequently, is that we go into new areas where our living space, we're constantly going into remote areas like rainforests to build and have a variety of things. We're going in there for people to live, to sample the environment, to study. I mean, the virus Zika certainly existed, but we didn't become into our lexicon until the World Cup and the Olympics in Brazil, because a lot of the venues were well into the rainforest, particularly some of the soccer venues. I'm a soccer nut. Go Man U. So all of these things are part of it. So going into the areas. The other ones that are interesting are what we classify, and I'm almost done, with resurgent or re-emergent diseases. The common factors. The two that are the poster children are clearly tuberculosis and measles. We'll get into vaccines later. The MMR, the measles, mumps, and rubella vaccine, has been an excellent vaccine. Measles may be the most contagious disease known, spread respiratorily. Everybody gets it. When I was a kid, I know I'm doing one of those. We all got, they called the usual childhood diseases. The only one I escaped was mumps, measles, German measles, rubella, chickenpox. I ended up, before my wife and I, as we were married, decided to have children, just in case I got just the mumps part of the mumps vaccine. I had to wait 3 months before we were interested in having children. Very successful vaccine. People have a tremendous amount of distrust today for vaccines. A lot of it came from an erroneous article we'll talk about that blamed it on autism. The British Medical Journal that published it retracted it with enormous apologies. This was about more than 20 years ago. showing that it was terrible research. Why they published it, I'll never know. The researchers drummed out whatever they did. I'm not telling you you're going to get my spin on vaccines. It's all about the preservatives. They were using a mercury preservative. Mercury is a neurotoxin. Talk more about it. But there are a lot of vaccines that are safe, and you can see, and we politicize them now, unfortunately, with changes from the CDC. I think we overused them previously. Now maybe we're tending toward underusing them, but that's a whole different lecture. Measles is what we see. Measles outbreaks. Measles gets everybody. My mom and all the other moms in our nice neighborhood at the end of first grade took us on a bus ride to New York City to go see the circus, which was at Madison Square Garden, the old Madison Square Garden, not the new one now, which was made in 1967. It's on 8th Avenue and 5th, if you're familiar, and 50th, if you're familiar with it. It was a walk from the Port Authority bus terminal. We all went there. It was effectively a measles and chickenpox party. We all got the measles, and a couple of weeks later, we all got the chickenpox. How's that? The moms, Ruth, you're in trouble. It's a good thing we didn't have 1-800 for a child. I would have called. Honest to goodness, I'm not. They did it all the time, and you wanted your kid to get a good dose of it because they knew that would stimulate the immunity. Now we have the immunization to do that, but because people have a distrust of vaccines, we're seeing outbreaks. Just look, say recent measles outbreaks. Google that, you'll be astounded. The other one is TB. We nearly had TB licked. The reasons: vaccine laxity. We don't use a vaccine for that, so it's not apropos here. There is a vaccine that's very ineffective in adults that's used in other countries. India would be the perfect example. If you want to travel there, you will have to get this vaccination. Understand now that the reason we don't use it, we will always test positive. We nearly eradicated disease with TB testing, chest x-rays, and treating when necessary. But because of immunosuppression, worldwide travel, aging populations, there's so many immunosuppressant drugs. They started with transplants when they became big. These are all causes. And we saw, and HIV particularly, because that was the early immunosuppressant, and we began to see outbreaks of respiratory diseases, including tuberculosis. Why we don't use it and why they use it in India, the single most common reason for TB, overcrowding. TB is not, and we'll talk about, we have a whole unit in respiratory diseases. It's not occasional contact. If somebody had TB in this room, and they coughed a couple of times, you're not getting it. Unless you were in that room constantly with them, and that's the trouble. Like a plane ride, for a long plane ride, you could have it. So, I mean, there's all sorts of issues associated with that. It's very interesting, but that's really, we nearly had it eradicated in this country. And now it has really surged. We're seeing a lot more problems with it. If you're going to clinical, you're going to see patients with TB. There's a lot of precautions because it's airborne. They're going to be in rooms where they're sucking the air out. It's like reverse isolation. Because they don't want to spread into the hospital. More about that down the road. I have a lot of great anecdotes about this. Thank you very much. I'm so happy everybody came. Most everybody, thank you, and have a great rest of your day. Friday, hopefully we finish this chapter. Hardly breathe.